Why compare proteomes? Of course, to study expression levels of normal versus disease cells. That is, for example, to identify targets and devise drug molecules as well. And how mass spectrometry can be used to relatively quantify proteins. What is the simplest procedure we can think of? So what we can do is simply we take three solutions from a normal versus a disease, and we do basically three bottom-up experiments for each of these samples, and we obtain protein IDs as well as quantities, and then basically do the statistics for each of the proteins that are there to get the normal versus the disease and identify if there is a significant change. I want to mention here that one of the fundamental assumptions and how we go about doing this experiment is that we start with the exact same number of protein amounts, the total protein amounts. That is based on the hypothesis that the total protein amount does not change significantly, for instance, if you treat with a molecule or when there is a disease state. However, these hypotheses can be altered depending on the problem that you are going to solve. The quantification, of course, can be performed using, again, extracted ion profiles of related peptides, or you can do spectral counting the peptide spectral matches, where I described how the experimental data is basically compared to the theoretical data with an associated protein. And this type of an approach is called label-free methods in quantitative proteomics using mass spectrometry. And I want you to pay a bit more attention onto these terminologies will be basically used in many articles. And for single protein based experiments and non-complex studies, appreciably reproducible data can be achieved using the label-free approach. However, for more complex samples, sometimes the extracted ion chromatogram reproducibility is low due to variability in LC-MS experiments, because you have to repeat the same experiment 3 times. But still can be used for very abundant and very responsive peptides during the LC-MS experiments.
All right, this is a classic question for you, Aruna.
Yes.
So why would you use mass spectrometry to check expression levels when there's Western blot and protein microarray? Is it because it gives more information?
Yes, it does give more information. I think Amber would explain this thing better than I would, but I'll try my best. So in a Western blot procedure, what you get is basically you analyze only one protein. It's like a targeted mass spectrometry approach. You are targeting one protein only. But when you do bottom-up proteomics experiments, you basically get thousands of proteins identified and quantified. So it is something like you are doing thousands of Western blots all together in one particular experimental procedure. If that answers easily.
Yeah, that's a great answer, Rita. I think the other things, just kind of building off what we were talking about earlier, that are good to talk about. One of the challenges with Western blots is that you have to make sure that you have a high quality specific antibody. And so that's really difficult to actually obtain. It's more difficult than what we might anticipate. And a lot of times we trust vendors to tell us that antibodies are good for Western blot, but obviously they are not unbiased in their assessments because they're selling us these resources. And so that's something that you really should consider when you're trying to do things in an unbiased fashion. So whereas Western blot is going to give you a measure of something based on antibody affinity, mass spectrometry can actually give you sequencing. In this case, when we're talking about label-free and some of the subsequent approaches that Aruna will talk about, you actually get sequencing, so you get identification of sequence. And so that is a much higher level of analytical precision than what you get with a Western blot. And finally, Western blots are also very sensitive to saturation effects. And so many of you that have worked with Western blots know that a lot of the software will give you an indication that you've reached saturation, but you can also really heavily saturate things if you expose on film. And that's something that we don't have to deal with in a mass spectrometer, partially because we have that dynamic ion filling that I talked about earlier. So we really avoid that saturation point, at least in that way.